Dr. Tala Navab-Daneshmand. Dr. Navab-Daneshmand is an assistant professor of chemical, biological, and environmental engineering at Oregon State University. Dr. Navab-Daneshmand researches the fate and transmission pathways for pathogenic and antibiotic resistant bacteria from wastewater systems to environmental reservoirs, as well as the design of engineered systems to reduce and combat contaminants of emerging concern. Dr. Navab-Daneshmand was a postdoctoral researcher in Switzerland and a postdoctoral fellow in Montreal, Canada, and received her PhD in environmental engineering from McGill University in Montreal, MS in environmental engineering from Tehran University, and degree in civil engineering, bachelor's in civil engineering from Amir Kabir University of Technology in Tehran. Welcome.
Thank you for the introduction. I'm going to talk about antibiotic resistance crisis and its relation to the agricultural reuse of wastewater and biosolids. Next, please. With the increase of population worldwide predicted to reach 10 billion by the year 2050, there will be agricultural demand increase. And therefore, as discussed in this panel, there will be need for alternative resources in agriculture in terms of irrigation or soil amendment. Wastewater and biosolids being rich in nutrients and can be used as alternative resources. There are regulations in effect by different regulations agencies for microbiological quality, for reuse of wastewater biosolids in agriculture. In the US, these regulations are set by the Food and Drug Administration and Environmental Protection Agency in terms of enteric bacteria such as E. coli, which is a fecal indicator bacteria. What these regulations do not include are the human health crisis of antimicrobial resistance. Next, please. What is antimicrobial resistance? When bacteria become resistant to antibiotic treatments, that's referred to as antibiotic resistance or antimicrobial resistance. Another term I'll use is multi-drug resistance, and that is when bacteria become resistant to not just one or two, but three or more classes of antibiotics. This is referred to in media as superbugs. Just to put it in context, in the US alone, according to the most recent CDC report, there are annually 2 million infections due to antibiotic resistance, causing 35,000 deaths. The widespread use of antimicrobial therapies during the COVID-19 pandemic is reported to have increased in some countries. And these increase in antibiotic treatments during the COVID-19 pandemics are generally due to co-infections and secondary infections. Therefore, the pandemic has potentially increased the burden of antimicrobial resistance. The burden of antimicrobial resistance is also disproportionately affecting low and middle income countries and communities. This is mainly due to the insufficient supply of clean water and sanitation infrastructure, but also due to high potential for transmission of pathogens in these communities. Wastewater and biosolids being the treated sludge, as discussed by Dr. Verbila, my colleague earlier, are one of the main reservoirs, recipients, and resources of antibiotic-resistant bacteria and their determinant genes. So therefore, next please, Therefore, one of the gaps in the knowledge that in my research group we are trying to address are the time variance of antibiotic resistance in wastewater. So how does it change with time? The prevalence and abundance of antibiotic-resistant bacteria and genes, and their fate in soil after wastewater irrigation and biosolids amendment. Next, please. To address these gaps, we performed a study in a greenhouse setting at Oregon State University, where we had these 2-gallon pots and we planted carrots from seeds. We watered some groups with water and some groups with wastewater effluent. We added soil amendments as in terms of biosolids to some groups, and we continued the study for 11 weeks until we harvested carrots. During this time, we collected weekly samples of soil, and we collected samples from the wastewater effluent, and at the end, at the harvest, we also collected carrot samples. And we tested all these samples for prevalence and abundance of antibiotic-resistant bacteria and genes. Next, please. So here, I just want to talk about the time variance of antibiotic-resistant genes in wastewater effluent. On the left-hand side, you see a figure where on the x-axis, there are the target antibiotic-resistant genes that we looked at. And on the y-axis, there is the normalized concentration of these target genes, normalized to 16S rRNA. We measured the concentration of these genes, and then we did statistical analysis to see whether there was significant time variances for these genes during these 10 weeks of the study. We didn't see a significant time variance between these genes, meaning the concentrations remain relatively constant throughout the 10 weeks. And these 10 weeks were between winter, late winter and early spring in Oregon, where there was a significant temperature change and a significant change in precipitation. However, those did not impact these concentrations. On the right-hand side, you see correlation matrix between the correlation between these target genes that we looked at. The red boxes are showing significant correlations that are highlighted by some stars. There are many correlations and many conclusions from this figure. However, what I want to point out to is the column on the right, INT-I1 gene, is an indicator of multi-drug resistance. So this gene was strongly correlated with 3 of the genes we looked at. And these 3 genes, soll1, soll2, and BLA-CDXM, could, because of their correlation with this multi-drug resistant genes, could also suggest being indicators of multi-drug resistance. This can potentially be used in forming policies and regulations for antimicrobial resistance, being an indicator for in policies. Next, please. In this figure, you can see the concentrations of total and antibiotic-resistant E. coli and enterococci, total on the top, and the bottom ones are antibiotic-resistant targets for E. coli and enterococci. It's a crowded figure, but what I want you to take from this figure is that if you can see, they're so overlapping that you can't see, but we have water irrigation and wastewater irrigation in solid and in dashed lines. And there is no significant difference. There was no significant difference observed between the concentrations of either total or antibiotic-resistant target bacteria when we use wastewater irrigation compared to water. However, when we applied biosolids to soil, those are the red lines with circle symbols, the concentrations of total and antibiotic-resistant E. coli and enterococci increased in soil, but it increased significantly. But what was interesting was that the antibiotic-resistant bacteria declined at a significantly faster rate than the total counterparts. And that's good news. So they're not persisting as as far as the total counterparts. However, we did see majority of multi-drug resistant enterococci in these soil samples. At the end of the harvest, we saw that carrots harbored E. coli, majority of which were resistant to ampicillin. Next, please. So at the end, I would just like to emphasize that antimicrobial resistance is an emerging human health concern that can potentially be exacerbated by the COVID-19 pandemic, disproportionately affecting low and middle income communities. With the need to reuse wastewater and biosolids in agriculture, we need to pay specific focus on this crisis and understand how it affects different practices and different policies. Next, please. And at the end, I would like to acknowledge Catherine Mays, who was a graduate student in my research group and led the studies we just discussed, my colleagues, and the U.S. Department of Agriculture for funding this study. Thank you.